Here's a short video describing a poster on magnetic electrospun microfiber scaffold assemblies, examples of the use for three-dimensional cell-based screening applications.
So here at Aurelio Bioscience, as a contract research organisation, we're interested in helping our clients to screen for compounds that are active in their assays. And particularly one of the things that we do is we do some phenotypic screening for people, and we're interested really in screening compounds in more physiological assays. So to this end, what we've been doing is that we've been trying to develop our own 3D models, because obviously cells in 3D produce responses that are different to those in 2D. The way we've been doing that is we've been doing that utilising a material which is called Electrospun fibre. And specifically what we do is we work with a company called the Electrospun company in order to manufacture this Electrospun fibre. And then what we do is we impregnate that with cells. We allow the cells to grow in, on, around the fibres. And then what we're able to do is then we're able to manipulate those in our cell culture. So what we're showing here is actually that we can manipulate these in terms of making them into different sizes, different shapes of Electrospun fibre. In this case, we've made some hexagonal ones that are approximately 100 micron by 100 micron by 50 micron thick. We try and keep the thickness of the fibre structures quite low, because obviously if we go over like 200 micron or more, we may well get cell death inside the structure that we form. So what we're showing here is actually an image which has been taken using a scanning electron microscope of actually two scaffolds which were side by side. Yeah, and what you can see is that the cells are basically, after 4 days, the cells are basically completely colonised the electrospun fibres, growing on them, around them, and effectively forming what appear to be sort of hamburgers. The other thing that I didn't mention earlier on was that in terms of the properties of the fibres, what we're able to do is we're able to incorporate iron nanoparticles into the fibres when they're being manufactured. Now that's really interesting. Why is that interesting? Because it means that we can move them around with magnetism. So then what that means is that we effectively have a culture of cells, a three-dimensional culture of cells, on a material that can be attracted and repelled by a magnet. And so what we're showing here actually is the process in more detail in this figure, whereby we can grow the electrospun fibre objects in a cell culture vessel, shown up here, in suspension. And then what we can do is then we can then put them into a one-well plate, and we can image that one-well plate, and then by using electromagnetism we can pick up those scaffolds and we can move them into a micro-well plate. Now that micro-well plate can be an assay-ready plate. So what that means is that we have grown in a homogeneous way the scaffolds all together, and then what we're then doing is then we're moving them into the plate for the assay. The other thing that we can do, which I didn't mention either, was that we can incorporate fluorescent dyes into the scaffolds. Yeah, and by incorporating fluorescent dyes into the scaffolds, what that means is that for high content screening, you have a contrasting colour upon which the cells are growing, so you can actually see the electrospun fibres in the high content image. Yeah, and in addition to that, we can utilise that almost like as a barcode. So what we could do in one well is that we could have blue scaffolds and red scaffolds, and we could utilise those in order maybe to put normal cells on blue scaffolds and abnormal cells on red scaffolds, and then by imaging them we could tell what the different populations are on each scaffold. The interesting thing as well is that we can transfect the cells whilst they're on scaffolds using liposuction or using electroporation, and showing here basically the fact that we can do pharmacology in terms of looking at responses from the cells whilst they're on scaffolds. Down here in this figure, what we've been doing is that we've been putting cells onto scaffolds, right? And then what that gives us the opportunity to do though is to change the paradigm of cell culture. So the paradigm of cell culture is typically your cells are adherent, they're stuck to the bottom of a plate, and you have to change the reagents that sit above them. But what we can do is that we can actually take the scaffolds out of the well. So we have an adherent monolayer which is on a material that can be removed from that well and put into another well. And so what we've done here is we've done some kinase screening with a technology from Promega called target engagement. And we have our cells adhered to our scaffolds, and then what we're able to do is we're able to soak the cells in either the tracer or the competing compound. But then rather than changing the solutions that are on top of the cells and possibly losing cells and possibly causing an increase in the variability in our assay, what we can then do is we can then take the scaffolds out of the wells, yeah, and we can put them into another well that contains either the tracer if we were trying to do on, or the competing compound if we were trying to do off-rate studies. And so we can do both on and off-rate studies in cells, right, using this technology. Okay, so in this figure here, what we're doing is we're looking at the differentiation of stem cells on scaffolds. Yeah. Now I know many of you do differentiation of stem cells in well plates, and many of you know, as indeed I do because I've done it, the difficulty in changing the media probably every day, every other day, trying to maintain the cells, not necessarily losing them off the bottom of the plate, and the process that you have to go through really in order to end up after 28, 30, 35 days with a differentiated culture. So what we've done is done something slightly different. So we've taken partially differentiated stem cells, right? And then what we've done is we've mixed them with a small volume of media. And then we've used a really small volume dispenser, in this case the Mantis from Foam Latrix, yeah? And what we've done is we've used that system to dispense very small volumes into precise locations on a piece of Electrospun material, yeah? So the locations are actually on the pitch of a 96 or 384 plate. So where you'd find the holes, where you'd find the wells on a 96 or 384 plate, okay? And we've dispensed them continuously into that location, in the hope that what we end up doing is we concentrate the cells in that one location, in and around those fibers, yeah. But then what we can do, you see, is then we can take that whole sheet of material, and we can put that whole sheet of material into a petri dish, right, or a flask, right. And what that means is if I have to change the media every day or every other day, what I just have to do then is I just have to aspirate all the media out of the petri dish or the flask and then put new media in. And by doing that, I'm actually feeding all the cells that are on that sheet or that piece of Electrospun material, yeah? And then what we do is at a particular point in time at which we think our colonies, our cells are mature and that we want to utilize them, then what we do is then we use a system to actually punch out, right, the locations where those cells are, right? We punch out the material from the sheet, right, much in the same way that you would punch holes if you were doing an A4 ring binder. We punch out the material out of those, out of the Electrospun fiber material, and we punch them into a well plate, yeah. And so we're going from that sheet straight into a well plate by making those punch out shapes, yeah. And what we're showing down here is actually, first of all, A is a bright field image of part of a scaffold, and B and C are actually high content images utilizing some fluorescently labeled antibodies in order to look at FOXG1 and PAX6 in B, and tubulin and C-tip in image C. And the idea really is that those FOXG1, PAX6 and tubulin and C-tip are actually markers of mature cortical neurons. And then what we've done over here in D&E is we've utilized a system from Protein Simple called WES, which allows us to do high throughput Western blotting to do a blot of both PAX6 and CTIP2. And you can see the bands that are consistent with demonstrating that these neurons that are on our scaffolds are mature cortical neurons. So what we demonstrated in this poster is the utility of this electrospun fibre material. Because it's attracted to a magnet, it means that we can move it around in culture and between well plates. Because we can add fluorescent dyes to it, it means that we can utilize those dyes either in high content imaging to act as a contrast, or alternatively almost to act as a barcode in order to say the blue one is normal tissue, the red one is abnormal tissue. And in addition to that, we've demonstrated the utility of being able to grow cells, both recombinant cells and inducible pluripotent stem cells, on our electrospun fibres. And in addition to that, what we can do is that we can transit and transfect cells that are on our electrospun fibres in order to express different receptors. We've also, but not on this poster, but we've also been able to demonstrate that we can actually cryopreserve cells whilst they're on electrospun fibre. So what this means is that it changes the paradigm somewhat in the way we do cell culture. So rather than thinking about cells which are like two-dimensional objects on a two-dimensional surface, i.e. the bottom of a flask or a well plate, what you can actually start thinking about is that now we can form three-dimensional arrays of cells, right, on an electrospun lattice, electrospun fibre lattice, that we can then start to manipulate in culture. So, thank you for listening.
Contact us if you would like a copy of this poster, or if you would like to learn more about our work with 3D cell culture technology. Follow our YouTube channel for more such videos.